What is Yom Kippur celebration? I know it's Jewish, but like it's like a New Year and I think that.
I do not remember. I feel like I ask that question every year.
Okay, so we've talked about membrane proteins, how they get there, how they're modified, types of amino acids they're made up of based on what they're doing and where they are, right? So we're building on previous knowledge of biochemistry. Now we're adding to it a little bit of cell biology, right? So how do these membranes function? How does facilitated diffusion actually work through a certain membrane protein, okay? So keep in mind there's simple diffusion and there's facilitated. Simple is able to go right through the membrane. Facilitated requires these protein channels and carriers. Somebody explain to me really quickly why that is. Why would you have to have facilitated diffusion for, say, sodium or water? Yeah.
Doesn't it have something to do with like the charge and then the like polar versus non-polar?
Yeah.
And the size would also affect it.
Yeah. So what categories have to be facilitated? Charged or uncharged?
Polar.
So polar versus non-polar. Yeah. So the polar things. What's the classic example of a polar molecule that we've talked about so far? Water. So yeah, so we had a quick little discussion and a little challenge on the quiz when I asked, you know, of these things, what can go through? And your book kind of goes out of its way to say, all right, look, here's the reality of it. Some water can go through the non-polar hydrophobic region of the membrane. Some can, right? So let's just get that out in the air. But is it at a rate that's compatible with life? No. So it's like, yeah, some can, but not in a way that's going to keep something alive. Okay, so that's why organisms have evolved the aquaporin, right? And osmosis through a water channel. So in this case, we're looking at just a channel, goes from high to low, shape doesn't change, whereas if we're looking at a carrier molecule, the actual shape of the carrier molecule is disrupted, so you get a shape change that facilitates the push of whatever's being transported. So if a protein binds that thing that's being transported, the atoms that make up that molecule, whatever it is, disrupt certain bonds, let's say hydrogen bonds between amino acids, resulting in a structural change. And once the thing is passed, and those interactions are restored, you get the original shape. Structure and function and evolution can literally be interwoven into any idea in biology, right? So on the AP test, they're going to most likely give you a problem about some sort of thing in life science that will require you to talk about structure function, literally throughout the curriculum. Okay, so it's not necessarily important for the curriculum, but I'll go ahead and just reiterate the difference. Channels going straight through without structural change for the actual protein channel. Carriers, you're seeing that change in structure, okay? So next class, we will perform some artificial cell experiments. So when I say artificial cell, I mean the membrane is not biological, it's plastic. So we have dialysis tubing. Does anybody remember dialysis tubing experiments? I think my group, we did them. I don't know if Mr. Gallagher did them. I know he has a demonstration where he has a giant Ziploc bag with really concentrated dye that diffuses through the Ziploc. But we will use dialysis tubing to set this up. So essentially that semi-permeable membrane, it's an artificial membrane that's selective for certain things, not necessarily based on the biochemistry of the solute that's passing in the membrane, because there is no membrane, but it's usually a size restriction, right? Size also plays a role in things that can get through. We'll talk about bulk transport here in a bit. But for the rules of this little demonstration, if you have water with various concentrations of solutes on either side of this artificial membrane, various things will want to happen and various things will be able to happen. So if I give you the following restriction, that solute, those purple spheres, they cannot cross the membrane because they're too big. The pores of that artificial membrane are too small to let it pass. But it lets a 3-atom water molecule pass. So water can undergo osmosis in either direction. It only goes in one direction, but it can go through the pore to either side. So just looking at this illustration, where will solute want to go? From right to left or left to right? From right to left, from high concentration to low concentration, we call that diffusion. Well, where will water want to osmose? Left to right. Now my question to you is why?
The concentration of water initially on the right was less than the concentration on the left.
Good. So I'm going to flip that a little bit to say, you know, osmosis, diffusion of water. Diffusion is high to low. It's higher on the left than it is on the right, so it'll go from left to right. Now, a lot of you may be thinking the water is going to go from left to right because there's more solute on the right side. Now, that's going to get you to the right answer, but if you say something like to balance it out, you'll have the wrong reasoning for it. So it's going to go from left to right, from high concentration of free water molecules to low concentration water molecules on the right. Not to balance it out, it ends up balancing it out in this system, but biologically the whole balancing out thing is tricky. We'll talk about that in a minute, but it goes from high concentration of water molecules to the right, and that's the osmosis of water. And if we fast forward it, we looked at this artificial system, you'd see on the right side, water levels actually going up and concentrations of solute being equal on both sides, but there being more of that solution on the right side. So that's kind of trippy for some students because what's happening to the water levels in that beaker on the right side? It's going up, so that looks kind of silly, right? So some students, like they can't put that in their mind that, well, it's just going to be more amounts of that solution on the right side. What does that mean about water osmosing from one concentration of solute to another? What does it create? So yes, you're right, it does create equilibrium, but my question is, why is it defying gravity and being pushed up? If you've looked at the formula sheet for AP Bio, you'll see a whole host of formulas, many of which we'll never use. They put them there, I want to say to like intimidate. No, it's not intimidation. It's to offer potential, right? We don't know what we're getting in May. We don't know what kind of problems they're going to give. It might encompass any of that. There's like geometry formulas on there, right? So, but you may or may not ever use that. And one of the things they put there are equations for water potential, solute potential, but I've never seen them use that. I've never seen a symbol for water potential arise outside of actual practice questions. So I'm not saying that it won't be a part of your test. We're not going to spend a whole lot of time on it here, but I do want you to understand that there's a pressure being placed over here, right? A pressure to the point where it can rise up that high on one side to reach an equilibrium, right? To have an equal concentration on both sides. Usually, you see these sorts of problems in these U-shaped tubes, right? Do you remember this from freshman year? So it's the same idea, maybe a little more logical because it's actual tube, but you know, I as the question designer can make it as complicated as simple as possible. I define what can go, what cannot go, right? Based on pore size for that artificial membrane, but it's the same idea. Can we have things, again, going in opposite directions based on concentrations? Could I put like a whole bunch of purple things over here that can move from one side to the other? And would they go from left to right? Sure they would, yeah. I mean, our cells, think about the amount of different biological substances that we transport, right? We have all kinds of molecules, all kinds of ions, so a lot of different stuff is happening at the same time. Okay. So working with these ideas of diffusion and osmosis, let's talk about tonicity. Okay, your book had mentioned it as what? Let's see if we read. So another synonym for describing the concentration of solutes in various solutions is
I don't know how to pronounce it.
osmolarity. Yeah. So osmolarity is another way of defining comparison of concentrations of solutes. Good. So I tend to use tonicity for this. You can think of osmolarity also. High versus low osmolarity. So, it's the same idea. If you have low osmolarity, low concentration of solute, lots of water. But let's talk about hypertonic, hypotonic, and isotonic. These Greek root words say something about those solutions. If you put hyper in front of something, what does it usually mean? Yeah. A lot of, fast, high, What about ISO?
Like a deficiency.
Um,
Oh, sorry, ISO.
That's a
Sorry,
decent example of-
sorry, Dr. Kinzer. I thought you said hypo.
Hypo, yeah. Hypo would be, yeah, sure. You see hypo as deficiency because that root is like below, less than, right? So hypothyroidism, right? But ISO means equal, right? So when we're looking at these definitions, what you're looking at is the definition of the solution surrounding the cell, okay? So if I say that this cell is in a hypertonic solution, what does that mean about the concentration of solutes in that solution when compared to the solute in the solution inside the cell? For hyper? Did you mean less inside the cell or outside the cell? Yeah. So if you have lots of solute in your solution outside the cell in a hypertonic solution, you can infer that the concentration of solute inside the cell is less. So outside it's hyper, inside it's hypo. What about if it's an isotonic solution outside? But it's the same, and if you have a hypotonic solution outside the cell, less solute in the solution outside the cell, higher concentrations of solute in the solution inside the cell. So cells have different ways of reacting to those tonicities, right? So this idea of osmoregulation, like how cells account for the effects of this, is what we're going to discuss next. So if you put a red blood cell into a hypertonic solution, according to the diagram, and using your knowledge of diffusion and osmosis, what would happen to those cells? Agree? Yeah, those sure do look shriveled. It's actually the formal terminology for it is crenation. They undergo crenation. Why is that? What is the reasoning behind that observation? Why is crenation occurring?
The concentration of solute outside the cell is higher, so water wants to leave the cell to go into the solution and therefore it should be good.
Good. So because the concentration of water inside the cell is higher than outside the cell, it will leave.
Dr. Kinzer, could I ask a clarifying question, please?
Sure.
So in a hypertonic, hypotonic, and an isotonic solution, the rate at which it enters, like the rate at which water enters and leaves, they're not the same, but the rates are constant, right? Like it's in equilibrium. That's what I was trying to ask.
So I would say that you're only in equilibrium in the isotonic.
Oh, okay.
Does that make sense to everyone? Yeah, so the rate of water entering and leaving is the same in isotonic solutions, which would be the middle scenario here showing yellow arrow in, green arrow out. right? So for hypertonic solutions, it's going to draw a lot of water out. In fact, that's the principle behind dehydration, right? Anybody in here ever make beef jerky? I haven't. Right, but if you were traveling on the Oregon Trail and you just caught a bunch of fish, but you weren't hungry for a lot of fish, but you wanted those fish later, what could you do to them? Salt them? What do you do? Draw the water out of the fish? Yeah. So now you have fish that lacks water and is less likely to rot, right? There you go. Just in case you find yourself in a situation. Yeah.
Hypotonic water is moving out of the cell, right?
Let's talk about that one now. So if you find yourself in a hypotonic solution as a red blood cell, there's higher concentrations of water outside compared to inside. My question to you is why? Why then would there be higher concentrations of water outside compared to inside? In other words, is there something else inside of a red blood cell, solute-wise, that would make it so? And the answer is yes. There's lots of stuff inside of cells. Like what? Hemoglobin, right? So all those proteins, but especially hemoglobin for red blood cells, right? Remind me the function of a red blood cell. Carries oxygen. So the protein that carries the oxygen is the hemoglobin. So it's chock full of hemoglobin actually, which ups the content of solute technically inside the cell, in addition to ions and all that other stuff. You know, there's not a whole lot of organelles inside of red blood. In fact, red blood cells are enucleated. What does that mean? They lack a nucleus, right? You can see that concave nature in the center picture with the isotonic state. They're concave because they lack a nucleus. They don't have a genetic code. So they're just constantly replenished. The enucleation allows them to fit through very small capillaries. Right? At some point in our vasculature, we have capillaries so small that these red blood cells have to like march through single file. So they can't be rounded because of the nucleus. That's an interesting aside. But yeah, the purpose of the cell is to transport oxygen. So we'll come back to that in a moment. But if you're in a very watery hypotonic environment, it's going to go into the cell. Potentially doing what?
Could burst it.
Swelling and bursting or lysing the cell. What does lyse mean? To destroy, chop up.
You're like breaking down the membrane of the cell, like lysis.
Yeah, what's a connection to a previous piece of information? Anybody know my favorite organelle?
Oh, yeah.
The lysosome, yeah.
Why?
Yeah, it's like a bonus question every year to see who's paying attention. I mean, the reason is I studied membrane trafficking of proteins to the lysosome in grad school, so I'm always like, what's my favorite organelle? Just to see who's paying attention for those 2 minutes. Lysosome, yeah. Okay, well, if in either case your cells crenate or lyse, why is that a bad day? Yeah, because then you're not getting oxygen and what else? Yeah, you would. Why? Here's my question, why? Why?
Cellular respiration.
That's all day. I've got like another 50 minutes.
Mitochondria.
Yeah, so why? Why do you need oxygen? Sort of previewing things to come. For cellular respiration. So unless that oxygen gets to what key organelle? Mitochondria to perform cell respiration, then you're in trouble when it comes to what key molecule. Because yeah, it's Mitochondria.
Adenosine triphosphate.
Yeah, you lack ATP, right? Okay. So if you're sent to the hospital for severe dehydration, you were found in the desert, they're taking you to the hospital because you're dehydrated. They're worried about your red blood cells, amongst other things. What are they going to give you? Should they hurry and give you a bag of water? I mean, you're really thirsty. We want to bypass the whole stomach and large intestine sort of thing. What would they do? They'd give you an IV for sure. Anybody ever have an IV?
On IV?
Yeah. Yeah. So they have these vans that go around, and they can offer you rehydration through an IV if you want to pay for it. How crazy is that? Right? Like it's like yeah, I mean,
You know,
Is the person like?
You know- no, you know what they use it for? I should pause this recording. It's for hangovers. Like if somebody's like severely hungover, they can get an IV, and it's like supposed to help. I wouldn't know anything about it. Okay. All right. So yeah, so we're good with that. Oh, by the way, what are they giving you? So we agree that that's not water in that bag. I mean, it is water, but is it just water? What else is it? Like
Electrolytes.
amino acids, or like what is a nutrient?
Sugars.
It is. I mean, it could be, but it's usually like a saline solution, right? So sodium ions, chloride ions. Why is that?
Because then you can increase the concentration outside of your red blood cell.
Okay. Do we want the concentration to be super high, or like what are we looking at? Do we want it to be hypertonic, isotonic? Yeah, you want it to be iso. You certainly don't want a bag of water, because that's going to be which of the three? You'll be hypotonic and your cells will burst, right? You don't want to dehydrate them, so it can't be like 20% salt, right? So you have to find that balance. And I'm not sure I'm not sure if they measure it on molarity or percentages, but I'm sure it's pretty low. Probably like, single digit percentages or even less. Yeah.
Is it possible, like, if you don't drink enough, like your blood becomes a hypertonic solution? You drink too much and then die because you have too much water.
Yeah. So you can drown without even going into a body of water. You can drown internally. Yeah. So you ready for something scary and alarming? So there have been several instances around the country of shock jocks on the radio holding contests for things, right? And so I distinctly remember a competition for like whatever level Xbox or PlayStation it was at the time. It was the new one coming out, and so this radio station had a competition. Like, we're gonna have a competition to see who can drink the most water without peeing, right? And so you have people like, oh, I'm out. And it got down to 2 people. And you think about this, like, unless it's I don't know. It's probably somebody like that really wants this for their kid, but can't go out and buy it. So they'll do anything to do it, right? And so they kept giving these people water and saying, you can't pee if you want this Xbox, right? So what ultimately ends up happening? Yeah, your bloodstream becomes hypotonic and your red blood cells start to burst. This person died, by the way.
They basically killed it.
Yeah, this person died through this competition for the Xbox.
Murder by water. Murder by water.
Like, how would you, like?
Yes, there are symptoms that start to arise before one would die from the lysis. I mean, they fought through it. They really wanted to win, so they started getting delirious, and I don't know what the actual symptoms are, but okay. So let's switch from an animal cell to a plant cell. Okay. Keep in mind that I normally have taught an evolution unit at this point. So I'm constantly throughout the year trying to draw connections from previous learning and evolution into the current lesson, and this is no different. So we're looking at a plant cell. There are two really important features when it comes to a plant cell and its evolution that help it with water retention and water transport with osmosis. So we have the same three conditions: hypertonic, isotonic, hypotonic. If you're in a hypertonic situation, again, you're dehydrating essentially by removing water. Water goes from high to low. What remains intact?
Cell wall.
Cell wall. So you see the cell membrane starting to what they call plasmolyse. So this is plasmolysis. The shrinking away of the cell membrane from the cell wall. But that cell wall, which is full of what polysaccharide again? Cellulose. The whole purpose of it is to remain fixed and rigid, right? So it stays there. The inside of the cell wall, the cell membrane starts to shrivel. It essentially becomes concentrated, probably viscous, and that cell is not going to make it, right? Isotonic, we see an equal exchange of water. That cell is probably pretty happy, but I would argue that it's not in its happiest state in an isotonic state. Why is that from an evolutionary perspective? Yeah. Plants need stuff like water to grow. What's the biochemical reactions that they need the water for? Again, a preview of things to come. Photosynthesis. So water provides electrons for photosystem 2. So it's evolved to be really happy in a very aqueous state, right? So those cells really want to be in a hypotonic environment, believe it or not. In fact, the whole purpose, and not the whole purpose, but one of the features of the cell wall is to act as sort of a retaining wall, right? Because that cell wants to swell and burst, but it's confined by the cell wall. Also, the large vacuole storage of water. So it'll fill up, it'll store, and it'll be happy to be in a hypotonic environment. Another real life application of this that you see. Any of you guys like asparagus?
Yeah.
Have you ever bought asparagus? Well, first of all, it's really expensive. Where do you find it? Don't tell me in the produce section. Know that. Like, where is it?
Oh, there you have like water.
Like, I know these. I know. I know it's by the lettuce in the produce section. I know it's like the first thing you see at Giant Eagle.
You have like little sprinklers down on it.
Yeah, first of all, they love to really sprinkle all of it. Why is that? Well, water comes into them. They look nice and plump and juicy and fresh, even if they've been sitting there for a few days, right? So water gets drawn in. So yeah, that's why you miss them. But you really, really want asparagus to be in this state, turgid state, right? Because who wants to buy like asparagus that's like leaning over, right? You want it to snap, right? When you cook your asparagus, what do? You take the bottom and you snap off a little bit of it to get just like the fresh stuff, right? So you put it in a water so that water goes in and it remains, snappy. Same thing with celery, right? Anybody like celery? Okay. Celery's kind of a unique flavor. Celery's a unique flavor, but let me tell you, would you eat celery if it was chewy? Thank you. So if you buy celery a day too early, you should take it home and put it where? In a cup of water in the fridge, right? So that it stays snappy. Okay. Okay, so here's another picture showing the information a different way. It's there for you in the presentation. We will, in 2 classes, perform the big AP biology lab for this unit. It's actually one of my favorites. It is the simplest. You could do this experiment at home, okay? It's a great experiment. We're not actually going to get to do it because of the restrictions in place, but I will model some of this for you I'll put you into virtual groups, give you some data, and you'll reason your way through the lab. But essentially what we do is we take core samples of various tubers, right? Sweet potatoes, Idaho russets. So I get to go to Giant Eagle usually and buy like a couple big bags of potatoes. Yeah, it's interesting because I get like the weirdest looks. And then people are like, "What kind of soup are you making?" I'm not. I'm actually bringing these to experiment. And they're like, "Okay, you may leave. Right this way to the door." Yeah, I'm like, I'm gonna perform experiments on these with high school kids. But it's actually a lot of fun, so we'll consider that. So this could be a picture of one of my plants. We like to euthanize plants in my house. That one on the left hasn't been watered in a little while, right? It's wilted, right? It doesn't look very happy, not having a good day. So you have to water your plants because otherwise they dry out and they're not able to bring water in, right? But if very quickly you provide some water, the water will go from high to low very quickly back into the plant. And for most plants, if you catch them early enough, you can save them, right? Why is it that you can save a plant even after nearly killing it by drying it out? Connecting previous ideas. Why are trees able to stay alive over the course of winter? So they might store some water in their large vacuoles, but that one, maybe he has a little left, but probably not much. It's been a while. And it goes back to this photosynthesis idea. Water, photosynthesis. What does photosynthesis create for a plant? Energy, but like be more specific. It's actually
sugars.
the molecule is glucose that then is used to make energy through what process? Respiration and what organelle? Making what product? ATP, right? So yeah, monosaccharides are energy, but really ATP is energy, right? That's what we're trying to get to. So plants have evolved a method for storing glucose. How do plants store glucose? Glycogen in their liver? No. Do plants have livers? No. How do they store it? As starch, right? So in many of these roots, there's large storages of starch. That's why we use the potatoes in the AP Bio Lab. What is a potato? It's a large ball of starch that we've stolen from the plant, right? They were saving it up for a rainy day and we've stolen it. And do experiments on it. Okay. So if any of this is not sticking, or if you're, having a hard time, you can always go back and watch some old resources on YouTube. I don't care what resource, you can go and find your own. My site has some. We will watch some of these. The point of this is don't be afraid to go back to some old resources that you may perceive as like a little more simplistic. It's helpful. We'll do some of these in class again, but more resources the merrier if you're struggling. Okay. Let's talk about a practical, real-life example of diffusion and osmosis. Anybody know what this is?
Paramecium.
Paramecium. Okay. Paramecium fall into what category of life? There he is.
Animal, like
Isn't it like an algae? It's algae, right?
Yeah, algae are also protists. He didn't have a poster, so. So if you're an organism, like a paramecium, you have to figure out ways to do things. And I don't mean like study. I mean like you have to evolve mechanisms that keep you alive, right? So endocytosis is one we'll talk about eventually. But let's talk about how they control for water, right? Because this organism lives in water. It has evolved these contractile vacuoles. So these are adaptations that allow this organism to contract the vacuole and to sort of wring out excess water inside of itself. So if it feels itself swelling a little bit, contractile vacuoles can actually pump it out of the cell, right? They are a cell. So my first question for you, if that is the case, what sort of environments, they said that they're aqueous, they're water environments, but tell me something about these environments. Hypotonic. So what bodies of water are hypotonic for organisms? Fresh water. Yeah, so freshwater ecosystems is probably where you'll find this particular paramecium, because fresh water relative to a cell that has stuff in it is very hypotonic. So water would go from the outside to the inside in order to maintain high fitness, that is to survive and thrive and reproduce. This thing has to figure out a way to get the water out. The way that it's figured out through evolution, an adaptation called a contractile vacuole. Okay, animal cells have vacuoles as well. Plants have those large storage ones. We have small storage ones. Its adaptation is contraction. You can squeeze and wring things out. Okay. Take a look at this prompt and tell me what you think. This is an example of a type of question you'll get from the College Board. Some people will read it and be like, he didn't teach me about paramecia. This guy's no good. What is this question about? About reading the graph, yeah, for the most part, right? But it's about relationships, right? And understanding that certain amount of energy to contract a pump might be more or less useful given the environment that the organism finds itself in. So, first of all, College Board needs to put a title. They didn't. Shame on them. They would have lost 2 points. They would have lost 2 points. Now, what did we say we like to usually graph on the y-axis? Dependent variables. So this thing depends on what you're studying. So it looks like the relative use of ATP by the contractile vacuole depends on something. What does it depend on? The environment. The relative salinity of the environment. So it depends on the environment. So what is the data showing us? What happens to the amount of ATP needed as the salinity increases? Yeah, so if you take a freshwater paramecium and you experimentally put it in various concentrations of salt, in a low salt concentration, it may still be taking on water and having to pump, so it might need a certain level of ATP. But as the salinity goes up and you become less hypotonic, more hypertonic, the use of that ATP will diminish. So what was your answer? Ooh.
D.
So we're all over the place though, but so you just agreed with that assessment, right? ATP usage is going down as salinity increases.
Yeah.
So that means increasing the salinity of the environment a little at a time until the ATP usage reaches the minimum, right? Okay. This is an AP type question, right? It's not like which of the following is hypotonic? You know? Okay. We're going to switch gears a little bit and move over to 2.9, the last section of this unit. You probably saw my message earlier. We now have assigned to you progress check 2. You have like 3 weeks to get it done. Okay. We're going to talk about mechanisms of transport. So what type of transport have we talked about for the most part thus far? Passive transport. Good. So this is passive. No energy required. Simply using what to allow things to be transported? The concentration gradients? Yeah. Yeah, diffusion through concentration gradient, right? So there's a whole host of things that must go on inside of a cell to keep the cell alive. It's very big, right? So we talk about homeostasis freshman year. And we're pretty much satisfied with homeostasis if you give us the generic example. Homeostasis is keeping things balanced. What does that mean? What comes to your mind when I say that everything is balanced? Equilibrium? Okay. Are all things at equilibrium inside of the cell or inside of our tissues? No, in fact, I would argue that in order to maintain homeostasis and balance, one must create situations that are not balanced. That is to say, you're creating concentration gradients for whatever this is on this side of the cell, whatever this is on this side of the cell. And in many instances, to create those concentration gradients, you have to go against the concentration gradient. What type of transport is that? Active. What does it require? Energy. ATP. Good. So if you really, really, really need to push sodium outside of the cell and bring potassium in to create concentration gradients for action potentials, which is literally linked to everything, you'd better be willing and able to make the ATP to do that, right? So we're going to cover some examples. The example that we'll primarily focus on is the sodium potassium pump. It's involved in the nervous system, the muscular system. So we're not going to focus on action potentials just yet. That's what they do. We'll cover that detail later. We're focusing on setting up an action potential with gradients. Electrochemical gradients, right? Because setting up a concentration gradient is one thing, but as soon as you get like a distribution of charge on one side of the membrane and on inside of the membrane, you're setting up the electrochemical side of it, right? So your book, provides this example of bringing potassium from the inside of the cell to the outside of the cell. We're going to talk about the sodium potassium pump. So what happens to membrane proteins when they work with ATP? So in other words, during active transport, if ATP is required, what usually happens to that membrane protein, like the sodium potassium pump? Max, take a shot.
They open and close to allow certain things to go from one side of the cell to the other.
Good. And opening and closing, starting out like this, going to this, and going back. Relate that to me using the idea that's like the common thread throughout this course. What happens here? Yeah, the structure of this protein, which is this way when not phosphorylated, changes to this when phosphorylated, and changes back when it's dephosphorylated. Again, it's throughout the whole curriculum. So let's watch a quick video of this in action.
Is a type of cell transport that requires input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred more common examples of captive transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites with 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is formed, releasing phosphate. This phosphate binds to the location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
Stop right there, amoeba sisters. Probably get hate mail for, I put that on YouTube, for going against the amoeba sisters. All right, let me turn on this camera real quick. Thea, can you see me? Are you okay?
Wait, Dr. Kitzer, were you talking to me? Sorry, I thought you said Mia for a second.
You're okay, you're okay.
All right.
I'm mumbling this mask, so.
No, yeah, I was able to see the video, yeah.
All good, man. Hey, so, let me see here. My bad. Yep, you're good. You're good. Okay.
Sounds good.
Yep. Okay, so I'm going to try to take care of him and take care of you guys at the same time. We'll go over these concepts. So structure, function, structure, function, structure, function, right? So the original structure of this sodium-potassium pump has it so that it's open towards the intracellular side, right? So we're looking at the protein in this conformation. It's shaped this way because of its primary, secondary, and tertiary structure. All the interactions between the amino acids, atoms of the amino acids, it gives it this shape. So it's open on the intracellular side. It has a high affinity for what when it's open to the intracellular side? The sodium, right? So 3 sodiums will bind to a certain part of this protein in this conformation. So it's open. They go in, they have a high affinity, they're there. There's also a binding site for ATP here. So over here, you'll see ATP bind, and ATP works by being hydrolyzed, right? So remember hydrolysis? What does hydrolysis do? Yep, it's gonna facilitate the breaking of bonds. So you're gonna lose your terminal phosphate of that triphosphate molecule, and that terminal phosphate remains where? Attached to the protein. So anytime you add atoms that shouldn't be on a protein, especially those phosphates, the phosphorus and the oxygen. What does it do to a protein? Changes the shape. So those atoms, and nobody knows how this works, I think, still. It'll break hydrogen bonds, and suddenly the conformation will change like this, so that now what was open on the intracellular side is open to the extracellular side. Also now, the sodium has a lower affinity for this protein. So they fall off because they're no longer as attracted to that protein. So it's going from this to this, right? And once it's in that state, it loses the sodium, and now it has a high affinity for what? Potassium. So the potassium is on the outside, it now finds its way in. It has a high affinity, it binds, and then you lose the phosphate. What happens when you're in this shape, this conformation, and you lose your phosphate? You go back to your original shape, right? So structure function. A lot of times students will ask, well, how does it know how to do this? And the answer is, it doesn't, right? It just does, right? It's like if it's open this way and you combine, you do. If you're open this way and you fall off, you do. And if you combine, you do. And it's constantly doing that. So it's very interesting how this all works. Okay. Any questions on that? So that's a really common example of active transport. Yeah.
In your reading, is it to the anaerobic cell is going to be negative, like overall, so that some, like, I forget if it's happening, but one of them wanted to go inside, yeah, because it had a lower concentration ingredient and also lower or negative ingredient.
Yeah, when we get to action potentials, we'll talk about depolarization. So the inside of the cell is depolarized, negative move charge, and that's because if you're pushing out 3 positive sodium ions for the two oxygen coming in, you're going to lower that internal charge, right? So that's setting up the depolarization. They're setting up that electrochemical region as well, with more positives being pushed out and coming in, but it's also setting up the concentration group. The first thing that happens, so it goes from high to low, outside in, right? And now you're it becomes a, we'll talk about potential down the road, but really the point here is to talk about protein shape. It's very neurosensitive. Okay. Yeah. Well, we do cover that too, like depolarization and then sodium rushing in and repolarization. We'll get to that. Are you interested in neuroscience? You just wanted to know.
I'm just curious.
Okay, yeah. What is sort of tough to take in there too is like, well, how is it getting negative if it's just positive things?
Yeah.
Is that what your question was?
Is there a reason I'm taking 3 of the sodium and only 2 of the
No, well, it's doing it because it can, right? And the biochemistry dictates that it happens. But the situation that it sets up is that internal negative charge. Okay, so you don't have to memorize any specific example of this, right? Your book mentioned all these different types of transporters. It was like, you know, here's a GLUT4 transporter, it transports glucose. You should know what aquaporins are and what sodium potassium pump is in this case, because those are the specific examples that we covered. But you might get any type of these, right? Generically, if you're looking at this, some are uniporters, some are symporters, some are antiporters. This depends on what's happening. So most likely the College Board will provide you with a background, some sort of scenario. And you just have to figure it out. You just have to see what's happening and use your knowledge of concentration gradients to figure out what's happening. So, you know, if you see 2 things coming in same way, symporter. If you see 2 things going in the opposite direction, antiporter. Okay. that's just a recapitulation. Here's an interesting example. So if you get sodium on the outside, where you have a concentration gradient and the sodium wants to come back in, what can happen is you have other molecules taking advantage of that concentration gradient in the flow to find their way against the concentration gradient, right? So here you have glucose hitching a ride, so to speak, through that concentration gradient. So we have to maintain concentration gradients to stay alive. We'll talk all about that when we get to cell respiration and photosynthesis, because they rely on concentration gradients too. Get out of your mind the fact that equilibrium is homeostasis. It's not. In fact, I would say, you know, this electrochemical gradients and, you know, creating gradients and going against the concentration gradient to create those, that's how we achieve homeostasis, to keep going, right? And this, the action potential is one thing, photosynthesis, cell respiration, those are all examples. Okay, real quick, before your quiz. Generically speaking, I want you to know of two forms of bulk transport. Bulk transport that brings things in, into endocytosis, and then bulk transport that brings things out. We've already covered this. That form is exocytosis. So the book offers specific sub-examples of this. So for endocytosis, if you're a single-celled organism like an amoeba or a paramecium, things that don't have mouths, how do you get nutrients and building blocks of life into your cytoplasm? So you grab it and you pull it in. So like for an amoeba, the way it eats is through a process called phagocytosis. So it's going to put its membrane around the thing it wants to internalize and undergo endocytosis. They call it phagocytosis because it's cellular eating. A specific example of internalization that I really enjoy because I studied it in grad school is this receptor-mediated endocytosis. Again, not something specifically you need to know for the course, but a really cool example of regulation. So if you only want these stars to come in when it's at certain levels, there are receptors that bind to that star, and only once a certain number of receptors are found does the internalization process happen. We see this inside of our retinas, which is how we recycle rhodopsin, one of the proteins involved in our visual system. So receptor-mediated endocytosis. Last but not least, exocytosis. Anybody know an example of exocytosis? We talked about it generically with the endomembrane system, putting proteins to the outside of the cell. What's an example? Since we're sort of thinking nervous system today, what's a parallel example? Neurotransmitters, right? So neurotransmitters that are dumped into the synaptic cleft to activate neighboring neurons. So those could be neurotransmitters. Dopamine, serotonin. Also happens with hormones. Okay. We good for a little quiz? Yeah. So stop and think, right? If you see an example here of something that's different or weird to you, stop and think about it and apply what you learned today. I remember. We believe in that. Oh, just enough.
Dear Dr. Kinser, are you going to send one through Google Docs?
Read my mind.
Great.